Beneath more than a mile of ocean, scientists discovered something that shouldn't exist. A real lake on the seafloor. It has a distinct shoreline, a visible surface, and even gentle waves, despite being completely submerged beneath the ocean. Yet, this isn't a normal lake. It's a deadly brine pool, a basin of super salty, oxygen-deprived water saturated with toxic chemicals, creating one of the most hostile environments on Earth. In 2020, the research vessel Ocean Explorer launched a submersible into the deep waters of the Red Sea. After 8 hours of surveying the abyssal floor, a featureless, almost completely lifeless plain of sediment and permanent darkness, the team was preparing to return to the surface. Then, the lights caught something, a shape approximately 20 meters across, dark, flat, sitting on the seafloor like a mirror laid face up in the mud. As the submersible moved closer, what had looked like glass resolved into something the crew had not expected and initially could not process. The surface was water, not solid, not optical illusion. A body of liquid resting inside the ocean with its own distinct surface and its own shoreline, where that surface met the seafloor around it. The sight was disorienting in a way that is difficult to describe from the outside. The submersible's lights illuminated a gentle, slow-motion wave lapping against a beach that existed at the bottom of the sea. A haze hung over the surface, a visible boundary layer between two different bodies of water, each with its own chemistry, its own density, its own rules. The team recorded everything. They were not the first humans to find something like this, but they were among the very few who had ever seen one in person. And what they were looking at was one of the strangest things on Earth. This is the story of the lakes at the bottom of the sea, where they came from, what lives on their shores, and why finding them changed everything. The first scientific record of a brine pool dates to a small article published on the 1st of March 1964 by a British oceanographer named Henry Charnock. The article describes what Charnock called anomalous bottom water, readings taken from instruments lowered 2 kilometers beneath the surface of the Red Sea that returned, results his equipment was not calibrated to interpret. The water at that depth was unusually warm, and it was extraordinarily salty, far saltier than the seawater above it. Charnock had no explanation for what he had found. He documented it, published the anomalous readings, and noted that by standard oceanographic understanding, what his instruments were describing should not have been possible. What Charnock had encountered, without knowing it, was one of the defining discoveries of late 20th century oceanography. His instruments had detected the surface layer of a brine pool, a body of water so dense and so chemically distinct from the ocean above that it sat separately at the bottom of the Red Sea, maintaining its own identity, its own temperature, and its own boundary, without mixing with the billions of liters of seawater pressing down from above. The observation was filed. The explanation would take decades to arrive. In the years that followed, similar anomalous readings appeared in the Gulf of Mexico and the Eastern Mediterranean. By the 1980s, the pattern was clear. These were real bodies of water sitting at the ocean floor, separated from the ocean above by a boundary as distinct as the surface between a lake and the air above it. The phenomenon had a name: brine pools. The discovery raised an immediate question that took several more decades to answer. Where does the salt come from? And what stops it from simply dissolving away into the ocean above? The existence of a lake inside an ocean seems to violate basic physical intuition. Pour anything into the ocean and the ocean absorbs it. The idea that a body of water could sit inside another body of water, with a clear surface between them, challenges the assumption that liquids always find their own level and blend together. The key is density. When salt dissolves in water, the resulting solution is denser, heavier per unit of volume than the fresh water it was added to. This is why the human body floats more easily in the sea than in a swimming pool, and why swimmers in the Dead Sea, where salt concentration reaches almost 10 times that of a typical ocean, sit on the surface like corks. The same principle operates at depth. Brine pool water is so heavily concentrated with dissolved salt, typically 5 to 10 times saltier than the surrounding ocean, that it is significantly denser than the seawater above it. Dense liquids sink beneath less dense ones. The brine settles to the lowest available point and stays there, collected in the natural depressions and basins of the ocean floor, unable to rise because the surrounding ocean is simply not heavy enough to displace it. The boundary between brine and seawater is not a solid barrier. It is a density gradient where the two liquids meet but cannot easily mix. A submersible crossing it feels measurable resistance, the dense brine pushing back against the hull. Instruments record an abrupt shift in temperature, salinity, and chemical composition within a vertical distance of a few meters. Below, the brine. Above, the ocean. Between them, one of the sharpest natural chemical boundaries on the planet. Why does the brine not gradually dissolve upward into the ocean? The answer is a process called double diffusive convection. Brine pools are warmer than the ocean above them, heated from below by geothermal energy. Both heat and salt want to spread upward, but heat diffuses approximately 100 times faster than salt. The heat escaping from the top of the brine warms the seawater directly above, causing it to rise and carry that heat away. The salt, moving far more slowly, stays behind. Cold seawater descends to replace what rose, reinforcing the density contrast that keeps the boundary sharp. The two fluids maintain the boundary between them in a self-reinforcing cycle that can persist for thousands of years. At the same time, the pool is being continuously replenished from below. Brine that escapes upward through the boundary is replaced by new brine rising from geological sources beneath the sea floor. The pool exists in a dynamic equilibrium, losing material at the top and gaining it at the bottom, that can be maintained for thousands or millions of years as long as the underlying geological supply continues. Some brine pools also flow across the seafloor in slow-moving rivers or cascade over seafloor ridges in underwater waterfalls, their dense liquid flowing like a heavy syrup across the ocean floor in complete darkness, carving channels in the sediment as they go. The geological story of where the salt comes from is as remarkable as the chemistry that keeps the pool intact. Brine pools have been confirmed in only 3 places on Earth: the Red Sea, the Gulf of Mexico, and the Mediterranean Sea. This geographic specificity is not coincidence. Each of these 3 water bodies shares a geological history that makes the formation of deep brine pools possible. A history involving ancient evaporation events that deposited enormous quantities of salt on the seafloor, now buried far beneath the present ocean floor. During the Jurassic period, approximately 180 million years ago, the Gulf of Mexico was sealed from the Atlantic by a narrow land bridge. The water evaporated, leaving a thick residue of rock salt across the basin floor. A similar event, the Messinian salinity crisis, isolated the Mediterranean from the Atlantic roughly 6 million years ago, depositing salt layers kilometers thick. The Red Sea has undergone comparable isolation events across its geological history, In each case, when the basin eventually reconnected with the ocean and refilled with seawater, the ancient salt remained, buried beneath layers of marine sediment that accumulated on top of it over the following millions of years. As more sediment piled up, the weight compressed the buried salt. Salt under pressure behaves plastically. Rather than fracturing like brittle rock, it deforms and flows, moving through the overlying sediment in columns and ridges called diapiers. These diapiers push upward toward the seafloor, and as they move, they allow seawater to seep downward through cracks in the sediment and come into contact with the ancient buried salt. The resulting solution, an ultra-concentrated brine, warmed by the geothermal heat of the underlying rock, is then squeezed back upward through the sediment and emerges at the seafloor, where it collects in depressions to form the pools that Charnock first detected in 1964. The lake at the bottom of the sea is, in effect, the ghost of an ocean that evaporated 180 million years ago. The geology explains the pool. What happens at its edges is where the story becomes extraordinary. The brine inside a pool is lethal to virtually everything that enters it. The salt concentration alone, 5 to 10 times that of seawater, is sufficient to cause immediate osmotic shock in any marine animal whose cells are not specifically adapted to it. When a cell from a normal marine organism is suddenly surrounded by liquid far saltier than its interior, water is pulled out of the cell by osmosis faster than the cell can compensate. The result is rapid cellular dehydration, the biological equivalent of having the water wrung out of every tissue in the body simultaneously. Fish and eels that accidentally enter brine pool water have been observed going into violent convulsions within seconds. Most do not escape. The brine compounds this lethality in several additional ways. It is anoxic, carrying almost no dissolved oxygen, because the density contrast prevents the mixing with oxygenated seawater that would otherwise resupply it. It is saturated with methane and hydrogen sulfide, gases toxic to oxygen-dependent life at the concentrations present in an enclosed, geothermally heated body of water. The combination of extreme salinity, oxygen absence, and toxic gas loading makes the interior of a brine pool one of the most hostile chemical environments on the surface of the planet. The bodies of animals preserved along the shorelines have been there for decades. The hypersaline water inhibiting bacterial decomposition in the same way that salt curing preserves meat. The shoreline of a brine pool is effectively an underwater mausoleum, its occupants frozen in the moment of their encounter with the boundary. Among the documented casualties at Brine Pool, shorelines are species rarely seen in the deep ocean at all: fish, crabs, lobsters, and eels that ventured into this part of the abyssal plain, drawn by the unusual chemical gradient the pool projects into the surrounding water. And that crossed the boundary before the consequences became apparent. The pool does not hunt, it simply waits, and the chemistry does the rest. For almost every creature that lives in the deep ocean, the brine pool is not a habitat, it is a trap. Almost every creature. The exception is the hagfish, an animal whose lineage predates the dinosaurs by over 300 million years. It enters the brine, feeds on paralyzed or dead animals, and returns to normal seawater. How it survives the osmotic and chemical conditions that kill everything else in seconds is not understood. Its physiology differs from most fish in ways still being studied. The research is ongoing. The brine kills, but at its edges, something remarkable is happening. An ecosystem that should not exist, powered by chemistry that science did not know was possible. The brine pool edge is not a uniform boundary. Between the lethal concentrated brine at the center and the normal deep ocean beyond the shoreline, there is a gradient zone, a region where brine chemistry dilutes and mixes with seawater in concentrations that, while not hospitable to most marine life, are not immediately lethal. In this zone, the chemical conditions are extreme by surface standards, but consistent and predictable. And consistency in an ecosystem is an invitation. At brine pool sites across the Gulf of Mexico and the Red Sea, researchers have documented biological communities of remarkable density. Giant Bathymodiolus mussels cluster in dense beds, individual animals reaching 20 centimeters or more, far larger than their shallow water relatives. Tube worms extend in colonies hundreds of meters long. Amphipods, squat lobsters, polychaete worms, and crabs move through the mussel beds. Larger predators, fish, octopuses, and eelpout visit to feed, using the mussel community as a food source the way large mammals use a water hole on dry land. In the otherwise barren abyssal plain, the brine pool shoreline is an oasis. What powers this ecosystem is not sunlight. No light of any kind has reached the brine pool since the ocean formed above it. The energy that sustains every organism in this community comes from chemistry, specifically from the methane and hydrogen sulfide that seep from the brine and the underlying geological formations. Bacteria living inside the gill tissue of the Bathymodiolus mussels convert these chemicals into organic compounds through a process called chemosynthesis. The same fundamental principle as photosynthesis, but using chemical energy rather than light. The mussels provide the bacteria with shelter and access to the chemical seep. The bacteria provide the mussels with nutrition. Neither can sustain itself without the other. The mussel bed is, in effect, a solar panel that runs on methane, an energy collector built from the very substance that makes the pool itself lethal. This was not the first deep-sea chemosynthetic community discovered. Hydrothermal vents found in 1977 established that sunlight-independent ecosystems could exist. Brine pools add a second category, driven not by volcanic heat, but by the seepage of ancient geological salt and its accompanying methane. Both systems demonstrate the same principle from different geological directions. Life can be powered by chemistry alone. The existence of life at brine pool edges changed the scientific understanding of where life can survive, and then raised a question that goes much further. The dominant origin of life hypothesis for most of the 20th century, developed from Darwin's 1871 suggestion and formalized by Brine and Haldane in the 1920s, proposed that life began in a warm, shallow tidal pool driven by sunlight, heat, and lightning. This primordial soup hypothesis shaped origin of life research for generations. The discovery of hydrothermal vent communities in 1977 introduced a competing hypothesis, that life began not at the surface but in the deep ocean, where chemically rich hot water provided a continuous energy source independent of sunlight and independent of the surface conditions that the early Earth's frequent meteor bombardment would have periodically catastrophically disrupted. The hydrothermal vent hypothesis has been the dominant alternative to primordial soup for the past 40 years, and it remains the most widely supported origin of life model in current scientific literature. Brine pools have added a third candidate, and the reason they are taken seriously is not primarily biological, but chemical. The early Earth's ocean, approximately 3.5 to 4 billion years ago, when life is believed to have first appeared, was chemically very different from the modern ocean. It was poor in oxygen, The oxygen-rich atmosphere that makes aerobic life possible today did not exist until photosynthetic bacteria had been producing it for hundreds of millions of years. It was rich in methane and hydrogen sulfide, produced by volcanic and geological activity at a rate far exceeding the modern baseline. And it was significantly saltier than today's ocean because the water cycle had not yet had enough time to dilute the salt leached from the young Earth's crust. These three conditions, low oxygen, high methane, high salinity, match the interior chemistry of a brine pool almost exactly. This is not a coincidence that goes unnoticed in the origin of life research community. If the chemistry of the early ocean resembled the chemistry of a modern brine pool, then the biological communities that have adapted to brine pool conditions may represent something closer to the original conditions of life's emergence than any other environment currently accessible to science. The bacteria living in mussel gills at brine pool edges, processing methane in the absence of oxygen in a highly saline environment, are not exotic extremophiles adapted to unusual conditions. They may be living in conditions very similar to those that existed everywhere on Earth when life first began. The brine pool is not an outlier. It may be a remnant. If life could begin in conditions like these on Earth, the question that follows is inevitable. Europa, one of the 4 large moons of Jupiter, first observed by Galileo in 1610, is covered in a shell of water ice. Beneath that ice, gravitational tidal, flexing from Jupiter's enormous mass, generates enough internal heat to maintain a liquid water ocean estimated at 100 kilometers deep, more than twice the total volume of all the water on Earth. The ice shell blocks all sunlight from reaching that ocean. The ocean floor, in contact with Europa's rocky interior, is likely geologically active. The combination, liquid water, geological activity, permanent darkness, is the combination that produces hydrothermal vents and brine pool conditions on Earth. Enceladus, a small moon of Saturn, approximately 500 kilometers in diameter, erupts plumes of water vapor, ice, and complex organic molecules from fissures at its south pole. Analysis of those plumes by the Cassini spacecraft, which flew through them multiple times before its mission ended in 2017, confirmed the presence of molecular hydrogen, a chemical signature strongly associated with active hydrothermal reactions between water and rock. The subsurface ocean of Enceladus is warm enough at the floor to drive chemistry. The ingredients for chemosynthesis are present in the plumes. Before the discovery of deep-sea chemosynthetic environments, hydrothermal vents, brine pools, and the communities that inhabit them, the subsurface oceans of Europa and Enceladus would have been considered almost certainly lifeless. Without sunlight, without photosynthesis, without the energy source that was believed to be the prerequisite for life, a cold, dark ocean under kilometers of ice seemed to offer nothing. The brine pool changed that assessment entirely. Sunlight is not a prerequisite for life. Chemistry is, and chemistry is available on both moons. NASA's Europa Clipper mission, launched in October 2024, is currently in transit toward Jupiter and is expected to reach the Europa system in 2030. Its instruments will analyze the composition of Europa's ice shell, map the subsurface ocean's extent, and search for evidence of chemical activity that would indicate whether the conditions for life are present. It will not be able to drill through the ice and sample the ocean directly. That is a mission objective for future spacecraft. But it will provide the first detailed picture of whether the ocean beneath Europa's ice shares any of the chemical characteristics that define the brine pool environments where life has established itself at the bottom of Earth's sea. A small article in a scientific journal in 1964. An oceanographer reading an impossible instrument, reading. A lake at the bottom of the Red Sea that should not have been there. From that beginning, a chain of discoveries led to the understanding that life does not need sunlight, that chemistry is sufficient, and that the dark ocean under kilometers of ice on a moon 600 million kilometers from the sun might not be as empty as it looks. Brine pools cover a tiny fraction of the ocean floor. They exist in only 3 regions of the world. They are lethal to almost everything that enters them. What they have revealed since Charnock's anomalous readings has been disproportionate to their size. That life can power itself on chemistry without light. That the conditions where life most likely began still exist and can be visited. And that the universe contains environments far more similar to the deep ocean than to any surface the sun reaches. The lakes at the bottom of the sea are small. What they contain is not.